Assalamu alaikum and Bismillahir Rahmanir Rahim. Dear student and viewer, today the topic of presentation is composition of blood. This topic is basically for BS Medical Laboratory Technology. My name is Tanveer Tara and I am lecturer at Sarhad University of Science and Information Technology. Student, this is our course outline during this semester, and it includes three different parts. The first one is basic knowledge about RBCs, basic blood and composition of blood, how blood is formed, erythropoiesis, its regulation, the formation of white blood cells, and the formation of platelets. Then we'll move forward toward anemia and how we can classify anemia and treat different forms. Then we have hemolysis. Hemolysis occur physiologically as well as pathological. We will discuss it in this semester. And introduction to WBC disorders, its morphology, and somewhat about leukemia, how leukemia is classified. And then we have WBC disorder like leukocytosis, leukopenia, its exact definition, and what are absolute counts. And then finally, we will touch platelets disorder as well. Okay, moving toward the content of this presentation, it includes physiology of blood, composition of blood, and function of blood, and the basic difference between serum and plasma, and what are anticoagulants, normal values of blood, that is the normal values of CBCs, and morphology of cell, which is very important. So let's move on. On this slide, I will try to discuss the physiology of blood. And if you know that blood is known as specialized body fluid. Basically, blood has been defined as highly specialized tissue, which along with the circulatory system is adopted to meet the needs of the body tissue and organ system. These needs include such function as exchange of gases, provision of nutrients, waste removal, and transport of chemical stimuli. So blood is composed of two phases, a liquid and a solid phase. Liquid phase is known as plasma, and the solid phase, or these are also called cellular components or cellular elements of the blood, that are red blood cells, white blood cells, and platelets. So from the previous slide, we came to know that blood is basically consists of two main parts. The first part is liquid part, and that is known as plasma. Its volume is about 55 to 60%, and the rest of the elements are formed elements or cellular elements, which composed of thrombocyte like platelets, erythrocyte that are known as red blood cells, and leukocyte that are known as white blood cells. So plasma may be defined as a fairly complex mixture of protein, electrolytes, and other chemical compounds which are dissolved in water. And the most obvious component of plasma is water, which is about 91% of the plasma. And the plasma contains different parts like water, that is 91%. It also includes electrolytes like sodium, potassium, and chloride. It contains different types of protein, like plasma protein. About 7% of the plasma is made up of proteins, and these plasma protein may be classified in a number of ways. According to their function, they may be divided into such categories as enzymes, and these enzymes inhibitors, transport proteins, complement factors, immunoglobulins, or also known as antibodies, and coagulation factors and fibrinolytic factors. So blood is composition of different types of items. Basically, liquid part, which I have discussed it earlier, and cellular elements that includes erythrocyte, leukocyte, and leukocyte further can be classified into granulocyte and agranulocyte. Like granulocytes, we have neutrophil, eosinophil, and basophil, and agranulocytes are lymphocyte and monocytes. The components of blood, the liquid components of blood is called plasma, and obviously it's a mixture of water, sugar, fats, and different types of proteins and electrolytes. The main job of the plasma is to transport blood cells throughout your body along with nutrients, waste products, antibodies, clotting proteins, chemical messengers such as hormones and protein that help maintain the body fluid balance. So the one part that is plasma is composed of different type of chemical substances that includes nitrogenous wastes, electrolytes, gases, fibrinogen, clotting factor, globulins, immunoglobulins, antibodies, albumins, protein, nutrients, amino acids, and a lot of different types of hormone that are included in the plasma, where the rest of the part that is formed element are known as cellular elements are basically divided into three different parts that are leukocyte, which is known as WBCs, white blood cells that fight against the infections, like neutrophil, which fight against bacteria, eosinophil, which rise in the allergic condition, and basophil, which also rise in the allergic or parasitic infection, and agranulocyte, that is lymphocytes and monocyte, and the rest of all are erythrocytes. So after summarizing whole blood, so the whole blood is composed of two parts. One is plasma, the other one is formed elements. Formed elements are only of three types, which I have discussed earlier, and rest of the blood is plasma, which also include in the blood. Overall, these all are known as whole blood. What is a whole blood? So a whole blood is that blood which runs through the veins, arteries, and capillaries. This is known as whole blood, and this whole blood is a mixture of about 55% of plasma and 45% of the blood cells, as previously I have discussed it, and about 7 to 8% of your total body weight is blood, and an average size man has about 12 pints of the blood, and an average size woman has 9 pints of the blood. So after knowing the composition of blood, now we have the curiosity that where do blood cells come from? So blood cells derived from hematopoietic stem cells, and what are these hematopoietic stem cells? They are basically formed in the bone marrow throughout the highly regulated process, and that process is known as hematopoiesis. Now hematopoiesis is a general term. Okay, hematopoiesis stem cells are capable of transforming into red blood cells, white blood cells, and platelets. As I told you that hematopoiesis is a general term, so whenever the red blood cells are formed throughout the hematopoiesis, that process is known as erythropoiesis. So the formation of red blood cell is known as erythropoiesis, and white blood cells, so the formation of white blood cell is known as leukopoiesis, as we all know that white blood cells are also known as leukocyte, and the formation of the platelet is known as thrombopoiesis. So these stem cells can be found circulating in the blood and bone marrow in people of all ages, in adults, in old ages, and even in the newborns in the umbilical cord they have present. So if we define hematopoiesis, so it is a specialized process. It is a specialized process of maturation, differentiation, and proliferation of all the cells. So after knowing the composition of blood, now we are going to present the different types of cellular elements, and the first one is red blood cell. Obviously it is very important function, and a lot of function red blood cells have. The most abundant cells in the blood is red blood cells, and its count is 4.5 to 5.5 million per microliter, and accounting for about 40 to 45% of its total volume. And the shape of red blood cell is biconcave disc with a flattened center, or in other word we can say that it's a biconcave disc, that is they are thicker at the edges than in the middle, and its size is about 7.5 micron. The diameter of the red blood cells are 2 micron thick at the edges. The essential role of erythrocyte is to transport oxygen to the respiring tissue and to carry carbon dioxide away from the tissue. This is a very important function. Red blood cells are ideally designed both in form and content for these tasks. Since the exchange of gases is the prime function of erythrocyte, it is clear a large cell surface area will facilitate this gas exchange. That's why it's a biconcave disc, and the biconcave shape allow a much larger ratio of surface area to content them. For instance, a round or globular shape, it is possible to design an even more efficient surface to volume ratio for the red blood cell by producing many folds and invaginations of the membrane. However, this shape would interfere with the smooth passage of red blood cells through the capillary blood vessels. The smooth round shape of the erythrocyte produces little friction as they are squeezed through the narrow capillaries tube. And red blood cells start as immature cells, which is known as erythroblasts, and then slowly and gradually after 7 days it becomes mature and released into the bloodstream. Like whenever it goes toward maturation, the first stage is erythroblast, then basophilic erythroblast, polychromatophilic erythroblast, arthrochromatophilic erythroblast, then reticulocyte, and finally red blood cell, that is mature cell. While discussing the red blood cells, we cannot ignore hemoglobin, because the hemoglobin has a very important role, and that is carrying oxygen from the lungs and then return back carbon dioxide. The color of hemoglobin blood appears because of the large number of red blood cells which get their color from the hemoglobin, and the percentage of whole blood volume that is made up of red blood cell is called the hematocrit, and is a common measure of the red blood cell, where we should know that hematocrit, also known as PCV, that is packed cell volume. And regarding these all, red blood cells are also ideally suited for their function, and each erythrocyte contain large quantities of hemoglobin. We have to know that a red blood cell contain a huge number of hemoglobin molecule, and that hemoglobin is the respiratory pigment that allow oxygen and carbon dioxide to be carried out in the red blood cells. Their shape, that is the shape of the red blood cells and content of the red blood cells, ensure an efficient exchange of gases in the tissue and lungs, but also the number of erythrocytes present in the bloodstream. And we have a number of erythrocytes in the bloodstream, that is 4.5 to 5.5 million per microliter. So hemoglobin has a very important role, and the most important role is carrying oxygen from the lungs to the rest of the body, and then it's returned back. We have another very important cellular component, that is white blood cells, and white blood cells are divided on the basis of granules in two different forms, that is granular form and agranular form, like granulocytes and agranulocytes, and these are neutrophil, eosinophil, and basophil. These are basically granulocyte, while lymphocyte and monocyte are agranulocyte. Their count is 4,000 to 11,000 per microliter, and these five cells are very important. They are fighting against the infection. Like we will discuss neutrophil. So neutrophil are the most numerous white blood cells and comprise about 55 to 75% of the total leukocyte population in the normal individual. They comprise 90 to 95% of all granulocytes. Neutrophil are easily recognized on blood smear, like we can see it on the right stand or Jimza stand or on any Romanowski stand. And the basically the second group of granulocyte is eosinophil. Because of the presence of many large red granules in the cytoplasm, they are known as eosinophil. And the main role of eosinophil is to fight against the parasitic infections like worm, specifically the larval form of the schistosomas or blood fluids, one of the major group of helminthes parasite. Other one that is granular cell, and that is known as basophil. They are named because their cytoplasm granules stain a deep blue black with the basic dye methylene blue, due to the fact that these granules contain many acidic molecules. And agranulocyte, they are lymphocyte and monocyte. A lymphocyte fight against the viral infection, while monocyte are the final group of leukocyte. They are the largest of the circulatory white blood cell, with a very high diameter, that is 17 to 24 micron, which is 2 to 3 times that of the erythrocyte, because we know that erythrocyte size is about 7.5 to 8 micron. So these five cells, these are white blood cells, and they have their own role. While we are discussing here just the introduction to white blood cells, not we are discussing here the detail of white blood cell. So it's enough for white blood cell that white blood cell have five different types: neutrophil, eosinophil, basophil, lymphocyte, and monocyte, and they have their own characteristic, and they have their own morphology, and they have their own function. Another very important cellular component is platelets, and platelets have also a variety of function. First, we have to know that platelets are not basically the cell, and why they are not fall in the category of cell, because they are not ideally cell. They are basically shredding of the cytoplasm, or they are the fragile packages of the cytoplasm, and they are very much smaller than the other blood cells. The average person has the count 1,50,000 to 4,50,000 per microliter of the platelet. Platelet are usually round or oval and have a very invaginated surface area, which allows for the adherence of many compounds. So they have very important role in sticking to the lining of the injured blood vessels and stop the blood, especially clotting factors. And they have the role for the primary plug formation. Platelets have a dual role in the circulatory system. First, they are instrumental in the maintaining the integrity of the endothelial lining of the blood vessels. When the number of platelets falls below normal, the endothelial cells tend to shrivel up, and as a result of the basement membrane surrounding the endothelium become exposed, thus promoting clotting. It seems that platelets play a nutrient role that allow the endothelial cells to maintain tight in the tracheal junction. And the second very important function is platelet have a role in the repairing any break in the vascular system, especially at the microcirculation level. That is where the capillaries, arterioles, and venules are found. Any break in the endothelial lining of the blood vessel will expose the underlying basement membrane, and platelets have a great affinity for this membrane, and when give a chance they will adhere to it in the large numbers. This starts the clotting process and will plug up the hole, and that is known as primary plug formation. So platelet has very important role, and platelet has certain role, but here in this topic we will just discuss its very superficially. So moving toward another topic, and that is anticoagulant. We will summarize these all, that basically blood is composed of two different parts. That is, one is liquid part and the other one is cellular element. The liquid part, that is known as plasma, which includes a variety of material, which includes electrolytes, protein, globulins, as discussed earlier. And on the rest, on the other hand, we have three different types of cellular element, that are red blood cell. Like we discussed red blood cells, its shape, why its shape is biconcave, and which molecule is inside the red blood cell, that is hemoglobin. And what is the function of hemoglobin? It's carrying oxygen and bring back carbon dioxide. Then we have we on white blood cell, that there are different types of white blood cells: neutrophil, eosinophil, and basophil, and they have their role. Then we comes toward the platelets, and platelets have also an important role. And why the platelets are not the cell, is we discuss it here. Now we are going toward the anticoagulants. So, what are anticoagulants? Is We can define anticoagulant as it's a chemical substance which inhibits the function, which inhibits the phenomena of coagulation. And in the laboratory, we are using different types of anticoagulants. Like we are using EDTA in the laboratory, that is known as ethylene diamine tetraacetic acid, a lavender sodium potassium EDTA. Its color is lavender or purple. This is strong anticoagulant, and these tubes are usually used for the complete blood count. Lavender tops, these tubes are generally used when whole blood is needed for analysis, and it can also be used for the blood bank procedures such as blood type and screening. We have another type of anticoagulant, that is known as heparin, which is used for chemistry and cytology as well. And we have anticoagulant like sodium citrate. Similarly, we have ACD, acid citrate dextrose, that is used in the blood banking, and CPD citrate phosphodextrose adenine. So these are different types of anticoagulants which are used inside the laboratory or blood bank. Earlier, we have discussed that blood is composed of two different types, like cellular elements and liquid form, and that liquid form is known as plasma. But in the laboratory, whenever we are discussing plasma or serum, so they have their specific definition. Like serum, what is serum? So when fibrinogen is removed from the plasma as a result of coagulation, such simply whole blood when centrifuged without anticoagulant is called serum. And whenever there is anticoagulant inside a tube and we centrifuge that, and the rest of the fluid we have, so that fluid is known as plasma. So this is the difference between plasma and serum. In the laboratory, we are treating plasma as in a sample. Okay, it's all enough for today. And if you have any questions, so we have a meeting on Google Meet. I will share the link with you people, and you will put your questions on a Google Meet, and inshallah I will try to answer them all. After summarizing this, so we have composition of blood in this topic. Composition of blood, like we divide the blood in two different form. One is liquid form, that is plasma, and the other one is cellular element. And we have the curiosity, that where blood come from, so that was hematopoiesis, then erythropoiesis, leukopoiesis, and thrombopoiesis. These are the formation, the proliferation, maturation, and differentiation of their specific cells. Then we have anticoagulant, that is it's a chemical substance that inhibits the phenomenon of coagulation. What is the difference between serum and plasma? So thank you very much for the listening. I will be inshallah with you in another lecture. Thank you very much.